Hi everyone! Today I'm going to talk about Lactobacillus acidophilus, which is one of the most well-known probiotic bacteria. You can find it in a lot of places: our gastrointestinal tract, the mouth, the vagina, and of course the familiar fermented foods like yogurt and cheese. It's a gram-positive lactic acid-producing bacterium, and it plays important roles in both human health and food production. So, what does L. acidophilus actually look like? Under the microscope, it has a rod-shaped or bacillus structure. It doesn't form spores, and it tends to appear either as single rods or short chains. One of its defining features is its thick peptidoglycan cell wall, which is typical of gram-positive bacteria and helps it withstand stress. As we know from lab, gram-positive bacteria show up purple after a gram staining procedure, so that helped inform the color choice for my artwork today. Many strains of L. acidophilus also have something called an S layer, basically a protein coat that helps the bacterium stick to host tissues and protects it from harsh conditions. On top of that, it has surface proteins that help it attach to mucus and epithelial cells, which is a big part of what makes it such an effective probiotic. Next, let's look at where L. acidophilus fits on the bacterial family tree. It's part of the Lactobacillus acidophilus group, which is a cluster of closely related species within the large Lactobacillus genus. Its closest relatives include L. johnsoni, L. gasseri, L. helveticus, L. crispatus, and L. amylivarus. These bacteria share evolutionary and metabolic traits, especially their strong preference for fermenting carbohydrates into lactic acid. They also tend to live in similar environments, like the gut or fermented foods. This close relationship helps explain why L. acidophilus shows up in so many probiotic formulas and food fermentation processes. Now let's talk about how this microbe eats and grows. L. acidophilus is homofermentative, meaning it mainly uses sugars like glucose and lactose and turns them into lactic acid. It's usually pretty nutritionally demanding. It can't make many essential nutrients on its own, so it needs amino acids, vitamins, nucleotides, and certain metal ions from the environment. This is why it thrives in nutrient-rich places like milk or the human gut. It's also very acid tolerant, which allows it to survive the stomach and live comfortably in low pH environments. Its metabolism changes depending on what's available, shifting gene expression to use whatever nutrients it has access to. This flexibility is part of what makes it successful in both the human body and fermented foods. One of the most important things L. acidophilus does is interact with other microbes. In the gut and vaginal microbiome, it competes with harmful bacteria for nutrients and attachment sites. It also produces lactic acid, hydrogen peroxide, and bacteriocins. These are natural antimicrobials that help keep pathogens like E. coli, Clostridium perfringens, Staphylococcus aureus, and Salmonella in check. It can even form clusters with itself and other friendly bacteria, helping it stick to surfaces and block pathogens from attaching. These microbial interactions are a huge part of its probiotic power. Beyond microbe interactions, L. acidophilus interacts closely with its human hosts and with the environments it lives in. On our mucosal surfaces, it helps strengthen the epithelial barrier, influences immune signaling, and contributes to an overall balanced microbiome. In food environments, it teams up with other lactic acid bacteria to produce the flavors and textures we associate with fermented dairy, while also making the food safer by keeping pathogens at bay. It's able to survive tough conditions like acidity and bile because of strong nutrient uptake systems and stress response mechanisms, allowing it to live in diverse ecological niches. As a symbiont, L. acidophilus provides several benefits. It supports immune regulation, reduces inflammation, and helps prevent infections by outcompeting harmful microbes. Its lactic acid production keeps the local pH low, creating an environment where pathogens struggle to survive. While L. acidophilus itself rarely causes disease, disruptions in its population, like from antibiotics or illness, can reduce colonization resistance and allow opportunistic pathogens to take over. Environmentally and industrially, it's also crucial for food fermentation, improving the safety, flavor, and shelf life of various products. Overall, its impacts stretch across human health, the food industry, and microbial ecosystems. Thank you for watching, and I hope this gave you a clear and approachable overview of L. acidophilus. My Zentangle artwork today is inspired by artist Lizzie Phoenix. That's her website if anyone wants to check her out. I also used this scanning electron microscope photo of L. acidophilus just as a reference image while I was drawing.
Have you ever heard how important it is to prepare and cook chicken properly, not to consume raw eggs, or have gone to the grocery store and the selection of spinach was completely empty? The reason for these warnings is due to a microscopic foodborne pathogenic bacteria known as Salmonella. This powerful little organism, about 2 micrometers in size, is rod shaped, covered in lots of little hair like protrusions, features multiple tail like structures, and has the ability to live in many conditions with or without oxygen. In more scientific terms, Salmonella is a gram negative bacteria that is a facultative anaerobe. It is bacilli shaped and a peritrichous organism, meaning it has multiple flagella around its body that it uses for motility. It is covered in fimbriae, short hair like structures, which are a very important part of the pathogen, as they play an important role in virulence, allowing the microorganism to adhere to its host cell or other environmental services such as food, stainless steel, and much more. Salmonella is mostly found in its natural habitat of intestinal tract of animals and humans. The main subtype of Salmonella is S. enterica, which is the leading cause of bacterial foodborne illness worldwide. And within the United States, it is the organism highest in incidence and morbidity. S. enterica originated from an evolution of E. coli. This took place through a combination of point mutations, horizontal gene transfer, and the acquisition of virulence factors. The variant that arrived between E. coli and S. enterica was S. bongori, which was restricted to and affected only cold-blooded animals. As a facultative pathogen, Salmonella can survive in many different environments, but is most commonly found living in the intestinal tract of animals and humans. It is within the intestinal tract that it finds most of its nutrients, feeding off carbohydrates and some amino acids such as aspartate. S. enterica may get its nutrients from carbohydrates, but it does not ferment lactose. It multiplies through binary fission after being ingested and invades host cells. Salmonella can survive in a broad range of temperatures, pH levels, and oxygen concentrations, making it a challenging pathogen to eliminate. Salmonella is not a friendly microorganism. Most of its interactions with other organisms are competitive and manipulative. It competes for iron, consumes other bacterial waste products, and creates inflammation to find nutrients. Being that Salmonella has such high tolerance, it can survive in many environments. Many animals are carriers, and a spread of Salmonella can start with animal feces contacting a water source, or simply human contact with animal skin. The bacteria can live in water, on plants, and inorganic surfaces it comes in contact with. It will also stay with a host like chicken or other livestock living in or on them. It is spread to humans through ingestion of improperly cooked animal products or unknown outbreaks on such plants as spinach. We can eliminate Salmonella by cooking animal products to a certain temperature, but it is harder to remove from raw vegetables as it does not just live on the surface where it is easily washed off. If Salmonella is ingested, a person can get severe stomach pain, diarrhea, and fever, which appear within 12 to 36 hours. In some cases, it can lead to hospitalization, and in severe cases, lead to death. With a simple case of salmonellosis, generally known as food poisoning, the recovery will take 4 to 7 days and does not normally need medical treatment. Diarrhea can last up to 10 days, so it's essential to stay hydrated during illness. Taking precautions to wash hands before and after food prep. Rinsing raw vegetables as best as possible and cooking animal products fully will help reduce the spread of Salmonella. Don't let Salmonella ruin your day. And here is my art that I created. It is a metal wire mobile featuring 3 little bacterial cells of Salmonella. You'll see here that it is gram stain for pink for gram negative. It has flagella, it's bacillus shaped, and it has fimbriae. Thank you.
Hi everyone. Today I'm going to be talking about Azospirillum. It is a genus of bacteria that plays a major role in plant growth and health. What makes Azospirillum interesting is that it isn't a pathogen and it doesn't form complex nodules on plant roots like other nitrogen fixating bacteria would. Instead, it works from the outside, living along the plant roots and quietly influencing plant growth in powerful ways. Because it connects microbiology to real issues like crop yield and soil sustainability, it's a great example of how important microbes are in agriculture. To start with, what Azopirillum looks like is what I have drawn here. So these bacteria are gram negative and they are shaped like rods. Sometimes they could be a little bit more curved. They have an outer membrane and a thin peptoglycan layer, and a periplasmic space. I try to represent that here with the thin peptoglycan layer and the periplasmic space. We have our circular DNA here. Um, and then one of the key features and what makes Azospirillum so unique is its way that it moves. So Azospirillum has a polar flagellum, which develop at the ends, and that is what they use for swimming. It can also develop, though, multiple lateral flagella, which are these guys on the sides of it. And that's what helps them move along the plant roots. And this motility helps bacteria reach what's called the rhizosphere. And what that is it is an active soil zone around plant roots where nutrients, like oxygen and plant chemicals, are constantly changing. Instead of entering plant cells, Azospirillum sticks within this root surface and forms biofilms, which give it the bacteria the sustainability and protection while they carry out all of its important functions. Speaking of important functions, the most important process Azospirillum is known for is going to be nitrogen fixation. This is how the bacteria convert nitrogen gas from the atmosphere into ammonia. This is a form that plants can use. So the enzyme is responsible for this is called nitrogenase, and it's made up of two main proteins that work together that use ATP and electrons that break the nitrogen triple bonds, which is extremely difficult to do. That's what makes this nitrogen fixation a high energy and specialized process. Nitrogenase can only function in low oxygen environments. So Azospirillum protects it by locating itself in microaerophilic pockets along the root and by lowering oxygen levels through respiration. Once nitrogen is converted into ammonia, that ammonia becomes available to the rhizosphere. And that is not only for the roots to absorb, but the soil and other microbes can use this to transform into different forms of nitrogen. So it kind of creates this nice little ecosystem down at the base of the plant root there. As far as Aspergillium goes, it belongs to the alpha protobacteria and includes a species. It includes a couple of species, sorry if I mispronounce this, Brazilianzine and Lipophorum. These bacteria have a circular chromosome and often carry a plasmid that contain genes related to nitrogen fixation. I tried to represent that here. I got a lot of ribosomes in the fluid here or in the matrix. We have my circular DNA there that helps promote all of those proteins for nitrogen fixation. Their closest relatives lives in environments where levels constantly shift, which helps explain why Azospirillum has adapted so well to the conditions nearest to the plant roots. Let's see. Beyond nitrogen fixation, Azospirillum also influences plant growth through other hormone production. It produces an 3-acetic acid, which is a plant hormone that increases root branching. So it also helps the plants develop more roots. And it also produces cytokines and gibberellin, gibberellins, which help the cell division and shoot development. And an enzyme called ACC deaminase that helps plants tolerate stress by lowering ethylene levels down there in the soil. So together, these affect all of these effects that Aspergillus has on the plant roots leads to stronger, larger root systems that take up more water and nutrients, thereby helping the plant grow more efficiently. So this bacteria also interacts with other microbes. Sometimes it competes for nutrients, but it also contributes to nutrient cycling by releasing ammonia. So its biofilm also allows it to coexist with decomposers and other bacteria that form small communities that influence how nutrients move throughout that rhizonosphere there at the base of the roots. In agriculture, Aspergillus has become a valuable biofertilizer. It's commonly applied as a seed coating or soil inoculant in crops like corn, wheat, rice, sugarcane, and various grasses. Um, Several studies have shown that plants treated with this Aspergillus biofertilizer they grow larger, they have improved nutrient uptake, and they produce high yields. One of the biggest benefits is that farmers can reduce their use of synthetic nitrogen fertilizers, which saves money and it decreases environmental population. Since Aspergillus doesn't require a specific host, plant, or root nodule, it is flexible and easy to use at a large scale, making it a leading microbial tool for sustainable farming. And then finally, Aspergillus is a great example of how small organisms could have a big impact on plant health and soil ecosystems. Its ability to fix nitrogen, support root development, and adapt to different environments makes it valuable in both natural and agricultural settings. Understanding microbes like Aspergillus helps us see how interconnected soil life really is and how these microscopic organisms influence the symptoms we depend on every day. Thanks for listening. My name is Joseph Martini, and I'm going to just give a little close-up of this guy here. Again, we have the polar flagella, the lateral flagella over here. We kind of have the inside of the cell here with its circular DNA, lots of ribosomes because it's performing so many different tasks and it needs all of those different proteins to perform those tasks to help make enzymes and things of that nature as well. And so I'm going to just hold it up to the camera there so you can see a little bit better. All right, and so that is my bacteria, Aspergillus. Thank you for listening.
For my project, I chose Streptococcus thermophilus. The anatomy of Streptococcus thermophilus is a gram-positive, non-motile, non-spore-forming coccus that typically appears as spheroid or ovoid cells arranged in pairs or short chains. The cells are approximately 0.7 to 0.9 micrometers in diameter and surrounded by the thick peptidoglycan cell wall characteristic of gram-positive bacteria. The thick cell wall provides structural rigidity and protection against osmotic stress. Some strains also produce exopolysaccharides that form a capsule or slime layer, an important factor because they contribute to the viscosity of fermented dairy products and enhance stress tolerance. Unlike pathogenic streptococci, S. thermophilus lacks many virulence-associated surface proteins, but possesses cell surface-associated enzymes and transporters specialized for the uptake and metabolism of lactose and peptides from milk. The cell membrane is rich in lactic acid transport systems that facilitate efficient fermentation during dairy processing. For phylogeny, S. thermophilus belongs to the kingdom Bacteria, phylum Bacillota, class Bacilli, order Lactobacillales, family Streptococcaceae, and genus Streptococcus. Within the Streptococcus genus, it is classified in the Salivarius group, which also includes S. salivarius and S. vestibularis. Phylogenomic analysis indicate that S. thermophilus diverged from its closest relative, S. salivarius, following adaptation to the milk environment, marked by genome reduction and specialization. For example, sequencing of early S. thermophilus reference strains showed elevated numbers of pseudogenes, loss of many genes associated with commensal or pathogenic lifestyles, and retention of traits beneficial for dairy fermentation. Analysis shows that important industrial traits such as bacteriocin production and oxygen tolerance were acquired from other lactic acid bacteria in dairy fermentations via horizontal gene transfer. Although S. thermophilus is closely related to the oral Streptococcus species, it is non-pathogenic and is highly specialized for life in dairy. Genetic comparison studies show that S. thermophilus forms its own distinct evolutionary branch, separate from harmful species like S. pyogenes and S. pneumoniae. These pathogenic species are able to expose proteins at their cell surface for cell adhesion and to escape the host defenses, while S. thermophilus has lost this ability. For microbial nutrition and growth, S. thermophilus is a homofermentative lactic acid bacteria, and among the primary organisms used in cheese and yogurt fermentations. It is thermophilic, with optimal growth occurring between 40 to 42 degrees Celsius and tolerating exposure to temperatures up to 60 degrees Celsius for less than 30 minutes. S. thermophilus cannot make several amino acids on its own and must obtain them from the surrounding environment. For growth, it uses peptides and amino acids released when casein is broken down and has specialized enzymes, such as proteinase and transport systems that help. In milk, strains that lack proteinase rely on cooperation with Lactobacillus delbrueckii subspecies bulgaricus to provide necessary peptides for dairy fermentation. The primary carbon source used by S. thermophilus for energy is lactose, which it transports into the cell with lactose permease. Lactose is broken down with beta-galactosidase into glucose and galactose. The glucose is used in the glycolysis pathway, which turns it into pyruvate and then into lactic acid through another enzyme called lactate dehydrogenase. This fermentation process produces ATP and makes the environment more acidic, which helps milk thicken and turn into yogurt or cheese. Some strains can use the galactose through special metabolic pathways, but others cannot and release it instead. In addition to lactic acid, S. thermophilus makes exopolysaccharides and small flavor compounds such as acetaldehyde and formate. These products work together to give yogurt their characteristic taste and consistency. From a physiological perspective, S. thermophilus has a stress response mechanisms typical of lactic acid bacteria, including tolerance to acid, heat, and oxidation. These adaptations include changes to the cell membrane and production of regulatory proteins that maintain cellular homeostasis under fermentation stress. These traits enable S. thermophilus to thrive in nutrient-rich but stressful dairy environments. Interactions of S. thermophilus with other organisms and the environment. S. thermophilus lives mainly in milk and works closely with other microorganisms, especially during dairy fermentation. In yogurt production, it forms a cooperative relationship with L. bulgaricus. S. thermophilus starts fermentation by quickly metabolizing lactose into lactic acid and formate, which stimulates L. bulgaricus growth. In return, L. bulgaricus breaks down milk proteins and releases peptides and amino acids that S. thermophilus needs for growth. This teamwork speeds up acid production and improves the texture and creates flavor compounds like acetaldehyde. Besides this partnership, S. thermophilus can also compete with other bacteria by producing bacteriocins such as thermophilins, which inhibit pathogenic microbes like Listeria monocytogenes and Bacillus cereus. It also makes exopolysaccharides that protect its cell, help it survive acid and heat stress, and improve the thickness and smoothness of fermented milk. From an economic perspective, S. thermophilus is the second most significant lab species with a market value of $40 billion.
Hey everyone, welcome to a talk about Clostridium botulinum. In 1793, in Weilbad, Germany, a sudden outbreak of deaths is traced back to the consumption of certain blood sausages. The mysterious illness is dubbed sausage poisoning. Later, February 4th, 1924, Albany, Oregon, in our very own state. A family of 12 all die after eating home-canned string beans in a salad. In the 1970s, Native Alaskan food preservation techniques are blamed after a series of deaths from eating fermented fish. It was later shown that this was due to fermenting in plastic rather than using traditional techniques. This is a reminder to trust indigenous knowledge. Meanwhile, in dark places with no oxygen, Clostridium botulinum thrives as an obligate anaerobe. Clostridium botulinum bacteria are ubiquitous in nature. They live in soils across the world. It's a reminder that it all begins in the soil. This humble Clostridium botulinum is responsible for producing the most lethal poison known to humans. Botulinum neurotoxin, BONT, is the cause of the often lethal condition known as botulism. BONT blocks neurons from communicating with muscles, leading to progressive paralysis which can stop one's breathing. Scientists suspect that C. botulinum developed its potent neurotoxin as a strategy to acquire a large food source, aka a corpse, for future generations. Typically, pathogenic bacteria are not rewarded by killing their hosts, but C. botulinum seems to aim to kill. Clostridium botulinum are rod-shaped, also known as bacillus, gram-positive, and usually, but not always, have paratrichous flagella for motility. Clostridium botulinum, like its other Clostridium cousins, has the ability to form endospores when under stress. This adaptation makes it extremely resilient to extreme environmental conditions and particularly challenging to destroy during food preservation. Zooming out a little bit, Clostridium is a bacterial genus in the Clostridiaceae family. It includes other infamous gram-positive anaerobic species like Clostridium difficile, causing diarrhea, Clostridium tetani, causing tetanus, and many others that regularly live among the human and soil microbiota. Clostridium botulinum is actually a set of at least 4 closely related but phylogenetically distinct species, 2 of which are known to cause botulism in humans. Scientists have wisely decided against renaming due to the potential negative impacts on public health communications. Therefore, Clostridium botulinum is used to describe any of the Clostridium species that produce the neurotoxin BONT. In addition to toxins, the bacteria secrete enzymes to break down complex proteins and chitin, which help to break down the tissues in a variety of corpses. Clostridium botulinum can then digest a variety of sugars, amino acids, and peptides via fermentation. Some common forms of human botulism. There are 3 main forms. The first is foodborne. Food preservation techniques can provide a perfect environment for Clostridium botulinum. Endospores are ubiquitous in nature and can easily colonize an airtight jar of beans or the anaerobic inside of a blood sausage. Once they start metabolizing, they produce plenty of BONT, which can be transmitted to humans upon eating. Sufficient heating will destroy the toxin, but not the protected endospore. Infant botulism is a poorly understood phenomenon in which a Clostridium botulinum spore germinates in the digestive system of an infant. It is theorized that the infant's underdeveloped microbiota allows for Clostridium botulinum spores to germinate, which typically cannot happen in an adult body. This leads to vegetative growth and the production of BONT. A rare form of botulism is found when an endospore moves into a wound. The main recorded cases of this happening are due to intramuscular injection of heroin, which is sometimes mixed with soil during processing. The bacteria divide using binary fission and can replicate rapidly inside a host. Common probiotics have been shown to outcompete Clostridium botulinum via resource competition, environmental acidification, and release of bacteriocins. This prevents the germination of spores and the proliferation of vegetative cells. The toxin produced by Clostridium botulinum has been developed for use in therapeutic treatments as well. Botox was developed as a usage of the BONT toxin to temporarily hide wrinkles by paralyzing facial muscles. A friend of mine, Molly, gets it to treat her migraines, too. Some references and acknowledgements. Thank you to Eli and Tai for their help with cutting out paper and transcribing some of these words. And as a side note, in Latin, botulus means sausage.
Hello, my name is Maham and I chose to do my project on Rickettsia. So Rickettsia is from the family Rickettsia. There are 4 genera. There's Rickettsia, Escherichia, Aurantia, and Cohexia. The genus I'll be focusing on is Rickettsia. The characteristics of Rickettsia are it is a small gram-negative bacteria. It is rod-shaped. It's very variable spherical bacteria. It is non-motile. It's an obligate intracellular parasite, and the outer membrane of the lipopolysaccharide is made of lipopolysaccharide with endotoxin activity. Its order is Rickettsialis, class of alpha proteobacteria, and phylum is from Proteobacteria. So how does Rickettsia actually get into the host? The bacteria infects an arthropod, which can be a tick, lice, flea, or mite that acts as a vector. The arthropod then infects a human and reaches the host cell. The bacteria can survive, grow, metabolize, and reproduce within the host cytoplasm. This is also where Rickettsia can receive ATP for cell function. Once the bacteria has caused significant reproductive damage, the host develops an illness. Rickettsia reproduces through the means of binary fission. When Rickettsia enters a host, they target smooth muscle cells and multiply by binary fission in the cytosol, which is the fluid portion of the cell cytoplasm, and causes heavy damage to the host cell. A reminder that binary fission is a form of bacterial asexual reproduction, where there is separation of a single microorganism body into two bodies, and therefore it divides genetic information into two parts. Metabolism of Rickettsia is a little complicated. Due to its intracellular pathogenic nature, Rickettsia can only survive in a host cell since it uses the host's machinery for metabolism and growth. Although Rickettsia has limited function of its own metabolism, where it may produce ATP through oxidative phosphorylation. In the host, Rickettsia uses the zipper and trigger mechanism to enter the host target cell. Rickettsia then uses the ATP-ADP symporter to exchange host ATP for bacterial ADP. Additionally, Rickettsia membranes are permeable to NAD+, which is a host energy source. On its own, Rickettsia uses glue oxidation to drive electron transport chain, which is coupled with oxidative phosphorylation in order to generate ATP. Since Rickettsia cannot use glucose as a nutrient, they oxidize amino acids and citric acid cycle intermediates instead. Rickettsia's relationship with other organisms usually consists of invading the host and starts host colonization. After entering the host, Rickettsia encounters host innate defense surveillance and may dodge many of them to colonize the host. The impacts of Rickettsia are pretty severe. These bacteria cause rickettsiosis, which are disease caused by Rickettsia, and most often cause acute febrile illness symptoms. The three most well-known illnesses from Rickettsia are Rocky Mountain spotted fever, epidemic typhus, and Murine typhus. Rocky Mountain spotted fever is caused by bacteria Rickettsia rickettsii and is transmitted from hard ticks among rodents and within rodents and humans. The patients present with fevers, headaches, and chills. Epidemic typhus is also called loose-borne typhus, which is mostly because the bacteria Rickettsia prowazekii is vectored by lice that infects humans. Patients are presented with high fever, depression, and rash. Finally, Murine typhus from Rickettsia typhi uses rodents as their main reservoir. The rat flea transmits bacteria among animal hosts to humans. These symptoms include headaches, abrupt fever, and chills. So I decided to do my art integration project, which includes a drawing of how Rickettsia infects and colonizes the host cell. In the first section, Rickettsia is in a tick parent vector. Through the transovarial transmission, which is the process of a tick parent passes bacteria to its offspring. The offspring now is the vector of Rickettsia. Rickettsia is replicated within the arthropod cell. In the third section, the arthropod has bit the human and has infected the human by breaking the skin barrier and is infecting the bacteria, and the bacteria infects the human cell. Rickettsia can now introduce itself into the host cytoplasm and cytosine, where it can reproduce cytoplasm and cytosol, where it can reproduce via binary fission. The cytosol of the cell is also the main area where Rickettsia receive host ATP. Due to this metabolic process, Rickettsia can survive, grow, and reproduce enough to cause disease to the human. Rickettsia is a very important bacteria for us to pay attention to since it causes significant human disease, and we should pay attention to them when we're traveling and when we are traveling in the United States in infected tick-infected areas so we can protect ourselves from acute febrile illness. Thank you for listening. Bye.
Hello, my name is Haley Metter, and a big reason that I got into dental hygiene is because it is one of the few areas of Western healthcare, at least, that is still preventative and focuses on self-care, which so does Akkermansia species, your preventative bacterial pal. When our oral microbiome is unhappy because of harmful bacteria that build up and cause plaque and calculus, as well as many other things, the gums become inflamed and bone is lost. But there are helpful bacteria that support and fight these symptoms, such as Akkermansia. This rod-shaped bacteria comes in at 0.621 micrometers. It is gram-negative, non-mobile, and non-spore forming. It is also anaerobic, and like most bacteria, it likes to be in the company of other bacteria and thrives. Akkermansia relies on mucin, which is a key ingredient of mucus, and it relies on this for its carbon, nitrogen, and energy source. The metabolites that are produced, such as short-chain fatty acids, a ketone, and polyamines, have their own benefits, such as glucose homeostasis, balanced metabolism, and they also feed not only the intestinal wall, but the bacterial community, which then that has their own ripple effect. Also, by degrading mucus, this supports the intestinal layer, the mucosal layer of the intestinal wall, keeps it balanced and strong to fight against pathogens. Even though this bacteria is found in 90% of adults, it was only discovered 18 years ago. And also because of its robust 16S RNA, its history is still not well understood, unfortunately, but because also most studies have focused on disease prevention, which makes sense. One of the most studied strains, though, is Akkermansia muciniphila, because it is very abundant, but it is also one of the Akkermansia strains that does biosynthesize B12 and cobalamin, which are major cofactors for many coenzymes. This also feeds its bacterial community, again, which is why it is such a symbiotic microbe. So, because it is also anti-inflammatory and fights bone loss, this makes it a key fighter against diseases such as periodontal disease. And periodontal disease is one of the leading causes of adult bone loss, so this is a very important bacteria, which is why it is now being considered the next generation's microbe. Not only that, but as we have seen, it doesn't directly affect the gut microbiota, but it does play a crucial role in fostering an environment that supports beneficial bacteria, and their metabolites contribute to the host's overall health, creating a symbiotic and happy home. While a probiotic supplement is becoming more of an option and will most likely be soon, that is an option for you, but you can always just focus on a healthy and well-balanced diet that include healthy fats, fruits, berries, legumes, leafy greens, and try your best to avoid sugar substitutes and overprocessed foods. And also visit your friendly, hopefully friendly, and local dental hygienist.
For more than 3.5 billion years, a remarkable lineage of life has shaped our planet. Cyanobacteria, our planet's ancient microscopic architects. These organisms are everywhere, thriving in places where few others dare to exist. in boiling springs, across sun-baked deserts, locked deep within polar ice, and drifting endlessly through the world's oceans. Wherever sunlight reaches, cyanobacteria endure, quietly influencing the world around them. Though small, each cell is a marvel of design. They possess a gram-negative cell envelope, are often wrapped in a protective mucilaginous sheath, and inside, networks of thylakoid membranes capture sunlight. They exist in a stunning variety of shapes and communities, as solitary cells, as clusters of colonies, or elegant filaments. Some are invisible to the naked eye, while others gather into mats large enough to be seen from the water's surface. During the day, sunlight becomes their power. Through photosynthesis, cyanobacteria convert carbon dioxide into life-sustaining energy, releasing oxygen as a byproduct. During the night, many cyanobacteria draw in atmospheric nitrogen and convert it into useful compounds. This process, known as nitrogen fixation, is essential for every ecosystem on Earth. In this remarkable rhythm, photosynthesis by day, nitrogen fixation by night, cyanobacteria quietly sustain the planet's carbon and nitrogen cycles. They belong to their own ancient phylum, Cyanobacteria, and are often mistaken for simple blue-green algae, but they are in fact sophisticated bacteria and the ancestors of one of evolution's most important innovations. More than 1 billion years ago, a cyanobacterium entered a larger cell and stayed. Over time, it became a chloroplast, the organelle responsible for photosynthesis in every plant we see today. Cyanobacteria have mastered survival in extremes. They flourish in geothermal springs, persist in frozen depths, and endure the harsh salinity of the seas. But their survival is aided by protective pigments and robust sheaths that shield them from intense sunlight, drying, and oxidative stresses. Today, they form the foundation of many ecosystems. In the world's oceans and freshwater systems, cyanobacteria are responsible for an enormous portion of global carbon fixation and oxygen production, sustaining life on a planetary scale. They also engage in intimate, ancient collaborations. In lichens, cyanobacteria supply energy to the fungal partners, who in return offer protection and structure. A stunning example of biological symbiosis. In dense communities known as biofilms, cyanobacteria trade nutrients, chemical signals, and even genes with neighboring microbes. Together, they form resilient networks that adapt to changing environments with remarkable speed. Humanity, too, has begun to recognize their potential. Scientists now cultivate cyanobacteria to produce biofuels, valuable carotenoids, and even technologies for carbon capture, harnessing their ability to turn sunlight and carbon dioxide into useful resources. But their power can sometimes cause disruption. In nutrient-rich waters, cyanobacteria may grow into vast and harmful blooms, releasing toxins that affect ecosystems and human health. These events are a reminder of their ecological importance and their extraordinary adaptability. They are ancient, they are resilient, and they remain one of the most influential forces life has ever produced. Cyanobacteria. Thanks for watching.
The bacteria that I will be covering today is Vibrio parahemolyticus. Vibrio parahemolyticus is the world's most infamous cause of foodborne illness. It is closely associated to shellfish, and it gives symptoms of acute gastroenteritis. When we think of illness related to oysters, it is likely from Vibrio parahemolyticus. In the following slides, I will cover morphology and phylogeny, as well as the main variables for survival, which include virulence factors, biofilm formation, and antibiotic resistance. I will also cover interactions with other microorganisms and the predator and prey relationships that they have. Vibrio parahemolyticus is a part of the Vibrionaceae family. Within the Vibrio genera, there are 40 species, and some notable relatives are Vibrio cholerae and Vibrio vulnificus. Vibrio parahemolyticus is gram-negative, bacillus, slightly curved, flagellated, facultative, anaerobe halophile. There are two different types, swimmers and swarmers. Swimmers have a polar flagella that allows them to swim through water, and swarmers have lateral flagella, which allow them to move through more viscous substrates. They are considered to be halophiles because they prefer to be in saltier water, about 20 to 25 parts per million salt concentration, and also warmer water as well. Some of the virulence factors that Vibrio parahemolyticus possess are multivalent adhesion molecule 7, or MAM7, which allows them to form biofilms. It is also a key component in cell-to-cell attachment, which they utilize for infection. They also utilize type 3 secretion systems and various gene-related toxins like thermostable direct hemolysin and TDH-related hemolysin, as well as antibiotic resistance that I will talk about in the next few slides. Type 3 secretion systems are essentially bacterial organelles that act as needles to directly inject bacterial proteins into a host cell. So in this image here, you can see a bacterial cell injecting itself with the T3SS into a different cell. These secretion systems are the primary mechanism of infection, and they affect the host cell's ability to produce energy. This ultimately leads to the loss of overall cell viability. TDH and TRH, or thermostable direct hemolysin and TDH-related hemolysin, are genes that enable Vibrio parahemolyticus to lyse the host's red blood cells. This essentially means just destroying those red blood cells. TDH is a pore-forming protein, so once the bacterial cell has infiltrated the host cell, TDH will come in and widen the pores on the host cell's mitochondria. This affects the host cell's ability to produce energy. TRH can change the host cell's morphology in a variety of ways, via disintegration of the nucleus or the cytoplasm, as well as external structures like microvilli. Another virulence factor of Vibrio parahemolyticus is antibiotic resistance. There are actually numerous antibiotic-resistant strains of Vibrio parahemolyticus that exist in the environment, and this is due to the misuse of antibiotics in human and also aquatic animal infections. This is important to consider because Vibrio parahemolyticus can transfer these resistant genes to other human pathogens. Vibrio parahemolyticus have a variety of interactions with other microorganisms. For example, it has a commensal relationship with zooplankton, which basically means that Vibrio parahemolyticus benefits from the relationship, but zooplankton is neither harmed nor benefits. Vibrio parahemolyticus utilize the plankton for protection and also transportation through the water column, and they're often called the hitchhikers of the bacterial world. On the opposite hand, we have Halobacteriovorax, which is actually predatory to Vibrio parahemolyticus. Halobacteriovorax occupies the periplasmic space of the host cell and uses the cytoplasm for nutrients until it can lyse out of the cell and spread more infection. Researchers are actually trying to figure out how to utilize Halobacteriovorax to minimize populations of Vibrio parahemolyticus. And here's my reference list. Thank you for listening.
Hi, I'm Neisseria gonorrhoeae. I'm a gram-negative Diplococcus obligate anaerobe bacteria. You've probably heard of me from your sex ed class, friends, or maybe even your partner. I'm an obligate human pathogen, which means I love people. I come from a big family, but only one of my siblings shares the same love for people that I do. Her name is Neisseria meningitis. She's the leading cause of bacterial meningitis. She's a little bit less serious about her love for people than I am, but she does seem to cause a bit more harm to her host than I do too. Also, people seem to find out about her faster than they do me. My other siblings in my genus are similar to me, but have other passions. We all like being in wet environments that are about 98.6 degrees Fahrenheit. We all enjoy being on animals' mucosal surfaces, and we are built to thrive in those environments. Before we get into my wants and loves, let's talk about my needs. I'm an obligate aerobe, which means I need access to oxygen to survive and thrive. For nutrition, I'm a little needy. In a lab, I need a special agar to grow. I do best on blood agar and chocolate agar. Yum. In the most basic terms, I need glucose, glutamine, thiamine, phosphate, iron, and carbon dioxide in order to thrive and grow. Now for my favorite topic, people. They've been obsessed with me for as long as I have been obsessed with them. A bunch of famous people have even talked about me, including Hippocrates, who I think is a pretty big deal. I'm even mentioned in the most printed book in history, the Bible. Each year worldwide, more than 80 million people find out that I am with them, and The Who even recently named me a superbug due to my obsession with humans and adaptability to their treatments. I usually find a new person to be with through the sexual contact of two people. You could say I love love. Once I find a new host, I bind to their epithelium in their throat, vagina, or rectum. When I first get there, I try to hide my best from the host body. Even though I love them, the immune system of my hosts don't really love me back. Depending on which zone I end up in, I use different techniques to hide from the body. In the cervix, for example, I bind to factor H, which the host body identifies as itself so it doesn't attack me. Once I get into a host, I use a few different proteins in my outer membrane called TDTSs that work to hoard some nutrients away from other organisms in the area, to both suppress the other organisms and pathogens and keep some resources for myself in case I need it later. Since I'm exclusive to humans, I had to learn how to adapt to avoid their rude medical advances and efforts to get rid of me. I use biofilms and horizontal gene transfer using my pili to share information with my peers and learn from their previous experiences with these things called antibiotics. Luckily, every time someone survives one of those series, we can share insider knowledge and become stronger, and build resistance to these horrible medications. Despite my efforts, scientists are still trying to figure out how to get rid of me. Once I make my new home in my human host, I'm contagious about a week after my first contact with them. Despite that, my host will still test negative for about a week after I'm contagious. That's still not long enough for me, but luckily it takes a lot longer for most people to find out that I'm even there. In female hosts, I typically fly under the radar. I usually do not cause noticeable symptoms in female hosts for a long time. Most find out about me through random routine screening at a doctor's office. I do cause some symptoms in male hosts, but it's usually just some discharge. Even though I love them so much, I can see why they don't like me. If I go undetected for long enough, I can cause some harm to my hosts. 1 in 5 females who do not receive treatment for me will get pelvic inflammatory disease, which can cause scarring and have long-term fertility effects, and requires a long course of medication to cure. Additionally, if I'm in a pregnant person, I can cause harm to the baby. I cause neonatal blindness and scarring and ulcers on the cornea of the infant. Thank you so much for taking the time to learn all about me.
Staphylococcus are gram-positive bacteria that are mostly found on human skin, nares, which is the nasal cavity, creases of the body, and other areas. Staph means cluster, and coccus means spherical. Each staph is about 0.5 to 1 micrometer. Staphylococcus are non-motile. It means they don't move around. They are non-spore forming, and they are facultative anaerobes, meaning they can survive with or without oxygen. Staphylococcus colonization in the human skin is completely normal, but if there's a high level of Staphylococcus in a broken skin, it can cause infection. There are common 3 types of Staphylococcus that are found in humans that can cause infections. These are Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. All of these 3 types are catalase positive. There are ways to differentiate the 3 types of Staphylococcus. Between these 3 types, Staphylococcus aureus is the only coagulase positive, and both Staphylococcus epidermidis and Staphylococcus saprophyticus are coagulase negative. Now, to differentiate the two, a novobiocin susceptibility test can be done, which is an antibiotic. If the bacteria is novobiocin sensitive, it is Staphylococcus epidermidis, and if it's novobiocin resistant, it is Staphylococcus saprophyticus. Staphylococcus aureus is usually found in the skin and the nose. It can cause skin conditions such as boils, folliculitis, cellulitis, and impetigo. Staphylococcus aureus also releases toxins. First toxin is hemolysin. This is a pore-forming toxin that destroys erythrocytes. Second, leukotoxin. These attacks the leukocytes and dies from necrosis. This causes the tissues to die and can lead to organ failure. Third, exfoliatin toxin causes staphylococcal scalded skin syndrome. Fourth, enterotoxin. These are generated when Staphylococcus aureus lands on food, and it can still be active even after the bacteria is killed by cooking. So this results in food poisoning. And last, toxic shock syndrome, or TSST1. These are produced at the site of the infection that can cause physiologic changes such as fevers, rash, decreased blood pressure, and organ failure that can cause death. Staphylococcus epidermidis is usually acquired in hospital, and it is a common cause of nosocomial blood infection. These infections happen to patients with prosthetic valves, cardiac devices, catheters, and the use of IV drugs. This is due to the exposure of the bacteria while the procedures are being done. Staphylococcus saprophyticus mostly colonizes in the perineum, rectum, urethra, cervix, and GI tract. This bacteria is the common cause of UTI, especially for sexually active females. Skin infected with Staphylococcus can be treated by topical antibiotics. Boils and abscess can be drained by healthcare professional. And if the infection is inside the body, it can be treated with oral antibiotic. And for severe cases, healthcare professional can also administer IV antibiotics to kill the bacteria effectively.
Today I'm talking about Yersinia pestis, the bacterium responsible for one of history's deadliest pandemics. Known for causing the Black Death in the 14th century, this gram-negative bacterium is still present today and remains a zoonotic threat. Yersinia pestis is a gram-negative, rod-shaped bacterium in the family Enterobacteriaceae. Its outer membrane protein, called AIL, helps it to attach to host cells and avoid destruction by the immune system. These features allow it to infect mammals, including humans, and cause severe disease. Yersinia pestis shares a close evolutionary relationship with Yersinia pseudotuberculosis, from which it diverged around 15 to 2,000 years ago. While Y pseudotuberculosis primarily causes mild gastrointestinal illness, Y pestis adapted to survive in both mammals and fleas, developing a much greater virulence. This evolutionary shift helped it spread rapidly and cause pandemics. Yersinia pestis is a facultative anaerobe, meaning it can grow with or without oxygen. This metabolic flexibility allows it to survive in diverse environments, such as flea guts or mammalian tissues. It also uses a specialized iron acquisition system to thrive in iron-poor environments, like the host's bloodstream. This system is critical for its survival and pathogenicity. The life of Y. pestis involves flea vectors and rodent hosts. Inside fleas, it forms biofilms in the digestive tract, which block the flea's ability to feed. This blockage causes the fleas to bite aggressively, transmitting the bacterium to mammals. This mechanism ensures that the bacterium's survival and spread through new hosts. Y. pestis affects ecosystems by occasionally reducing rodent populations, which can disrupt predator-prey dynamics. For instance, outbreaks in prairie dog, Cynomys lodovicianus colonies in the Badlands of South Dakota highlight the need for flea control and rodent monitoring to prevent larger epidemics. In humans, Y. pestis causes 3 types of plague. Bubonic plague, this causes swollen lymph nodes or buboes and has a mortality rate of 30 to 75% if left untreated. Pneumonic plague infects the lungs, and septicemic is very rare but is a blood infection and is almost always fatal without treatment. Prompt medical intervention, such as antibiotics, is essential to save lives. Y. pestis has several virulence factors that make it highly effective at causing disease. The EIL protein enables it to resist immune defenses, while the plasminogen activator enzyme breaks down host tissues, allowing the bacterium to spread rapidly. These features help Y. pestis invade, evade, and infect with deadly efficiency. Historically, Yersinia pestis caused devastating pandemics, such as the Black Death, which killed an estimated 25 million people in Europe. Today, it remains endemic in certain areas, with occasional outbreaks. This persistence reminds us of the need for ongoing public health measures to monitor and control this pathogen. In conclusion, Yersinia pestis, a pathogen of historical and modern importance. Its adaptability, ability to evade the immune system, and survival strategies make it a remarkable microbe. And by studying Y. pestis, we continue to learn about combating infectious disease and improving public health. I made an art project for our assignment. I researched classic ornament and historical pictures and images of the plague, and put together a collage of monoprints using an acid transfer or an acetone transfer pen. And it was really fun, even if it was such a sad topic to work on. It was really great to make something. And I wanted to do it. I did the printing on the back of sewing pattern tissue paper because I wanted the conceptual idea that this touched so many bodies.
Have you ever felt like this bear relaxing on the river? Well, there are significant issues you could be dealing with if there's Shigella in the water. Shigella is a gram-negative, non-spore-forming, non-motile, bacilli-shaped bacteria. Shigella is a chemoorganotroph and a facultative anaerobe and is often cultured on mac agar, which helps differentiate it from others based on its inability to ferment lactose. This characteristic is often used in lab settings to help differentiate Shigella from other bacteria such as E. coli, which can ferment lactose. Numerous gram-negative bacterial pathogens such as Shigella and Salmonella employ the T3SS mechanism to infect hosts. Upon contact with the host, the T3SS inserts its translocon pore into the host membrane, creating a channel for the delivery of effector proteins into the cell. You can see that here on this diagram. Transmission of Shigella often happens with dirty water, hand-to-mouth situations, and often in food handling. These bacteria have rapidly acquired resistance to multiple antibiotics through horizontal gene transfer, including quinolone resistance. Addressing Shigella infections requires a multifaceted approach, including improving water sanitation, monitoring antibiotic resistance, and enhancing public health infrastructure to prevent outbreaks. Let's try to keep our water clean so we, like this bear, can enjoy all that nature has to offer.
Captain of all these men of death, a zine about Mycobacterium tuberculosis. Throughout human history, one microbe has led to more disease and death than any other. We've found evidence of it on mummies from Egypt and Peru. The ancient Greeks called the disease it causes phthisis. The ancient Romans referred to it as scrofula. This disease is mentioned in the Hindu Rig Veda as Yakoma. In the 19th century, consumption developed a reputation as a romantic disease, responsible for the deaths of young creatives in their prime, like Charlotte Bronte, Frederick Chopin, and John Keats. This disease has been portrayed many times in stories, songs, movies, visual art, and even video games. We know this disease as tuberculosis, caused by the bacterium Mycobacterium tuberculosis, aka MTB. First identified by our friend Robert Koch, Mycobacterium tuberculosis is an acid-fast, gram-positive bacillus and an obligate aerobe. MTB's cell wall contains mycolic acid, a fatty acid, and a lipid polysaccharide complex called lipoarabinomannan. These give MTB a waxy coat that makes it resistant to physical and chemical damage. As we will see, this enables it to survive under remarkable circumstances. Tuberculosis has 3 phases of infection. During the primary infection, Mycobacterium tuberculosis is transmitted by droplets and inhaled into the lungs, where areolar macrophages phagocytize MTB. MTB releases lysosomes that destroy the macrophages and digestive enzymes that break down lung tissue, creating its signature tubercles. At this point, the immune system keeps responding. Usually, MTB is contained and the tubercles heal, and most often, this is the end. But sometimes, Mycobacterium tuberculosis takes advantage of its special ability to go latent. It hides in the lungs, just waiting for an opportunity, a time when the body's immunity is low. Then it undergoes reactivation. Reactivation causes pulmonary tuberculosis, and this is the version most of us know. The lungs are overtaken by tubercles. Signs and symptoms of pulmonary tuberculosis include a progressive cough, fever, weight loss, and the bloody sputum that we see in movies. Left untreated, patients usually die in 2 to 5 years. For thousands of years, we had no cure for tuberculosis. Then in 1943, Albert Schatz, a PhD student working with Selman Wakeman at Rutgers University discovered streptomycin. In 1945, a woman named Patricia was treated with streptomycin and her tuberculosis was cured. But Mycobacterium is persistent. Mycobacterium tuberculosis very quickly developed antibiotic resistance. And today, most cases of tuberculosis are treated with a lengthy course of multiple antibiotics. This example here is a 4-month course. It's the shortest I was able to find. But, of course, it's not that simple. As many as 1 in 3 people today are infected with M. tuberculosis. HIV and COVID have increased susceptibility to tuberculosis worldwide, and antibiotic resistance has given rise to multi-drug resistant tuberculosis, as well as totally drug-resistant tuberculosis. Mycobacterium tuberculosis kills 1.5 million people annually, more than any other infectious organism. TB is the leading cause of death for people with HIV. It contributes to increased antibiotic resistance worldwide. Right now, we have no simple solutions. The story of M. tuberculosis is as old as humanity, and it's not over yet.
For this presentation, we're looking at the microorganism Helicobacter pylori. Helicobacter pylori is a gram-negative, non-fermenting, non-sporulating microaerophilic bacteria. It's about 2 to 4 micrometers long with a diameter of about 1/2 to 1 micrometers. It has a helical shape with about 2 to 7 Lophotrichus flagella at the end. That means that all the flagella are used for movement in one direction. It does come in a couple other shapes, but the other shapes are not as useful, just in rods or curved. The spiral shape is the one that's most successful and impactful. Helicobacter pylori is within the bacteria domain, and as you can see on this slide, it shows the phylum, class, order, family, and then the genus and species, which is Helicobacter pylori. This microorganism is a highly successful and prevalent bacteria. I saw a variety of different estimates in terms of the prevalence, but one that I saw is approximately 2/3 of all people in the world are believed to have been infected at some point, with higher prevalence in the developing world. The vast majority of people who have this bacteria are asymptomatic, but those who do have symptoms can develop chronic gastrointestinal symptoms. H. pylori is believed to be linked to approximately 89% of gastric cancers, 5.5% of all cancers worldwide, making it the only bacterium linked to development of any cancer. H. pylori has extensive genetic diversity of strains, which helps it thrive. So within one person, there can be a lot of strains, and between different people. The genus Helicobacter consists of over 20 recognized species, with many awaiting formal recognition. In addition to a lot of diversity within humans, there are related Helicobacter species that have been identified in the stomachs of other wild and domesticated mammals. There are a number of features that allow this bacteria to survive and thrive in stomachs. It's microaerophilic, so within the stomach it only needs a little bit of oxygen, and it contains hydrogenase, which it uses to oxidize hydrogen in the surrounding environment to get its nutritional energy. From that hydrogen, it produces oxidase, catalase, and urease. The urease production is key to its survival because it takes it in, breaks it down, and it creates ammonia and carbon dioxide. That ammonia in the surrounding area increases the pH, making it more basic, less acidic, and creates kind of a window of less acidity right around the organism, which allows it to live. When people have an H. pylori infection, it impacts the entire microbiome in a person's stomach. It can be around 40 to 90% of all the microbiota when it's present. So it reduces the overall diversity. The change in pH impacts the ability of other microorganisms to survive. And then another way it can impact the stomach is that eradication therapies can also further ruin the microbiome. It uses its helical shape and flagella to burrow into the mucus lining of the stomach. So it gets through the mucus and up to the skin layer. Once it's in the epithelial layer, it produces adhesins which allow it to stick there and not get washed away. Once it is up against the stomach wall, it stays there and produces ammonia, which decreases acidity. The ammonia allows it to stay there, but the ammonia also is toxic to the epithelial layer, so it ruins this stomach lining. It creates an immune response, more acids. So basically, it sits there against your stomach wall, and then your stomach acid ruins your stomach lining, creating ulcers. With an H. pylori colonization or infection, people can experience abdominal pain, bad taste in the mouth, nausea, heartburn, belching, and the infection is related to a number of diseases: chronic gastritis, peptic ulcers, gastric adenocarcinoma, gastrointestinal reflux disease, and cancer. H. pylori has been isolated in saliva, gastric mucus, dental plaque, and feces. It's transmissible through oral route or fecal oral route. Right now, there are no vaccines, so hand hygiene, access to clean water are really important. There are a number of different antibiotics that can kill an H. pylori infection or colonization. Other therapies like proton pump inhibitors, bismuth salt, and a number of different foods are recommended. And if you're having all of the symptoms in the previous slide, it might be worth going to a doctor and getting checked out.
Hello, my name is Izzy Murray, and I wrote about the bacteria Treponema pallidum. Just some quick facts about Treponema pallidum. It's a sexually transmitted disease that causes syphilis. It's also a gram-negative bacteria. It's spirochete in shape, meaning it's very motile with important flagella that move it counterclockwise and clockwise. It's microaerophilic, which is important because it can grow without oxygen. It doesn't need a lot, but it does need some to survive. And it also must be in a human host, so it can't live outside of one. I also wanted to bring up the phylogeny of Treponema pallidum. It's kind of an enigma of how it all started, especially because it's increasing in rapid numbers, especially even today. So the first one is the pre-Columbian hypothesis, where it talks about how treponemal diseases in general over time and how they respond differently depending on changes to the environment. So that can mean a lot of things, whether it was temperature or climate. And then the other one is the Columbian hypothesis, which deals with Christopher Columbus and the spread of syphilis. The reason why people think that this is one of the huge parts of the spread was because of what was documented between two physicians who talk about syphilis, and then dating back skeletons from that time period was found to have syphilis-like lesions on them. So yeah, there were these two physicians that talked about syphilis and how it was kind of spreading everywhere and then spread to the US from Spain. Another thing I wanted to talk about was the stages of syphilis. So it's kind of broken down into 4 stages: the primary, secondary, latent, and tertiary. While syphilis is treated with penicillin, when it gets into the last 2 stages, it's kind of unlikely to be resolved and is usually just stays in the body forever. But the primary stage is categorized by enlarged lymph nodes, and then usually a lesion in a genital, oral, or anal region, kind of wherever that contact was transmitted. Usually the lesion is painless. And then the secondary stage is kind of the classic stage for symptoms, or at least one that most people have heard about. It's the rash on the palms of hands or the soles of feet. And then usually this is also associated with a fever. And then the latent stage is the chronic one. It's less likely to be resolved here. This is also a huge part where congenital syphilis can happen, which is syphilis being transmitted to a fetus from a pregnant woman if she has the infection. And then usually at this time, the fetus would be born with syphilis. And then the tertiary stage, the last one, this is the neurological damage one. It also can impact other organs, but it's most known for impacting the neurons function. Another note I wanted to mention was the historical impact of syphilis in medical ethics. So the Tuskegee syphilis study was a 40-year study starting in 1932. It examined 600 African American men in Alabama, and about 400 of those people had active syphilis, while the 200 were control. These men thought that they were being treated for Treponema pallidum, but the researchers were lying to them in order to see the effects of Treponema pallidum in the human host long term. This is, of course, a very horrendous study, and it did become a gateway to protecting human research subjects and ethics for future medical experiments, and it is mentioned in medical ethics courses today. I also wanted to talk about my art component, so I decided to make a puzzle with little spirochetes on it. I made it a puzzle specifically because, like I was mentioning before about the phylogeny, it's kind of hard to know exactly how syphilis spread. There's ideas, but it's kind of an enigma. They're also pink to represent them being gram-negative, and the white background is to envision them under a microscope. I also drew some of them, if you notice and look closely, as little snakes, because today we kind of look at syphilis and spirochetes as like little snakes. They're complicated, they can be wound up and kind of have that corkscrew shape. I don't know if anyone's ever seen the little plushies of bacteria or viruses, and syphilis is represented as a snake, so I thought that would be kind of fun to add that in here. Anyways, thank you for listening.
E. coli is the most studied and best understood bacteria to date. There are nearly 1,000 known strains of E. coli, with new ones still being identified. Commensal E. coli lives naturally in our GI tract, aiding us by taking up space that could otherwise be taken up by more pathogenic microbes and even other strains of E. coli. But even this commensal E. coli is capable of causing UTIs, sepsis, meningitis, and various enteric diseases, as it is an opportunistic pathogen. Other strains have varying virulence factors. E. coli is a gram-negative. It does not produce endospores. E. coli is rod-shaped and appears solo or in pairs. It may be motile with the presence of pernacious flagella. It is a facultative anaerobe. E. coli belongs to the Enterobacteriaceae family, like Shigella, Salmonella, and even Yersinia pestis, which cause the plague. It is acid tolerant and can survive in the pH of stomach acid for hours on the way to the intestines. E. coli is found in the mucus and epithelial cells of the intestines and make up to 1% of gut bacteria. Metabolic requirements are flexible, as E. coli has the ability to switch pathways when nutrient availability changes. Pathways such as glycolytic pentose phosphate, TCA cycle, and fermentation are some of the pathways. The source of nutrients of commensal E. coli in the biofilm are shed epithelial cells, dietary fiber, and mucosal polysaccharides, but each pathotype has specific nutrient needs based on the host microenvironment. E. coli can conjugate with other microorganisms, exchanging genes and ever-evolving. This is done by extending appendage called pili, making contact with another bacterial cell and transferring genetic information. But the most frequent way that E. coli exchanges genes is by bacteriophages, viruses that infect bacteria specifically. Some of the pathotypes of E. coli are enterohemorrhaginic E. coli, which produces a Shiga toxin. It is able to survive through innate host defenses like saliva and stomach acid because of its acid resistance. Once in the intestines, the Shiga toxin causes chaos, resulting in bloody diarrhea, renal failure, dehydration, fever, and fatigue. Enteropathic E. coli doesn't produce toxins, but it does attach interface microvillus. It causes persistent diarrhea and is a leading cause of death of infants in developing countries. Enterotoxigenic E. coli causes diarrhea by secretions of enterotoxins. Enteroinvasive E. coli is similar in many ways to Shigella, and this strain causes severe watery diarrhea, fever, and cramps. It may be local in the intestines or systemic. E. coli is found in the feces of animals, and the transmission of bacteria may begin with fecal matter contaminating food directly or contaminating water. Contamination that reaches water may be used in agricultural irrigation or reach waterways that lead to recreational waters and beaches. The USDA and FDA have guidelines to reduce transmission, which include inspecting facilities and testing food. The most common food contaminated are fresh produce, raw dairy, and undercooked beef. But it's not all so bad. E. coli has been the model of choice in the laboratory since the 1940s. And microbiology, as we know it, wouldn't be here today without E. coli.